Hello, welcome to today's lesson on the electron transport chain. The electron transport chain is the final and most important step in cellular respiration because it is a step that eventually now makes the ATP that is necessary for support of other metabolic processes in the body. Respiration involves about 3 steps. It involves glycolysis. There's also the citric acid cycle and the electron transport chain. All these processes make up the respiration, the process of respiration. However, ATP is the one that finally makes the energy that is needed in form of ATP, and it does so by the process of what? Oxidative phosphorylation. So it requires the presence of oxygen to generate ATP. And it occurs in the mitochondria. That's the site where this process occurs. It occurs in the mitochondria. So this is our electron transport chain. Now, what does it involve? As I mentioned, so this is the mitochondrial membrane. This is the mitochondria membrane. You have the outer membrane there, the intermembrane space, then the inner membrane. Then you have the inner mitochondrial matrix. This is where this process occurs. It has got about 5 proteins in form of what? Complexes. 4 complexes. The first one, as you can see, this one in green, we call it complex 1. Then this is complex 2. Then this is complex 3. This is complex 4. And this is another channel protein that is embedded with the enzyme ATP synthase. So all these processes, these proteins perform various functions. Some of these proteins have got channel pores that allow for the movement of protons from where? From the mitochondrial matrix into the intermembrane space. So now let us begin by looking at what happens at complex 1. So what really happens at complex 1? So complex 1 is also known as what? NADH dehydrogenase. So it receives NADH. NADH is a high energy compound. So it receives NADH. This NADH donates 2 electrons that are captured by complex 1. But in the process, as it receives the 2 electrons, it pumps the protons on the intermembrane space, this side. It pumps them, from the inner mitochondrial matrix, it pumps the protons into the intermembrane space. So complex 1 pumps about 4 protons from the inner mitochondrial matrix into the intermembrane space. It pumps 4 protons. Again, in summary, the NADH that comes to complex 1 donates 2 electrons. So these 2 electrons, when they are captured by this complex gets energized. When it gets energized, it opens up the channel that allows the pumping of what? Protons from the inner mitochondrial matrix into the intermembrane space. Then the number of protons that are pumped from the inner mitochondrial matrix into the intermembrane space is what? 4, about 4. That is complex 1. So complex 1, also known as NADH dehydrogenase. Then complex 2, which is here, complex 2, which is also known as the succinate dehydrogenase. Succinate dehydrogenase. This one receives electrons from what? FADH2. This is another high energy compound that gets reduced. So NADH goes to complex 1. FADH2 goes to complex 2. So when it goes to complex 2, it also donates 2 electrons to energize complex 2. but as it donates 2 electrons, it is converted to FAD, just like NADH is converted to NAD plus after donating the 2 electrons. Now, the difference at this point is complex 1 pumps 4 protons into the intermembrane space. Complex 2 does not pump any protons into the intermembrane space. It just gets the electrons and gets energized. Then both complex 1 and complex 2 donate their electrons to complex C to coenzyme Q. Coenzyme Q, which is also known as a ubiquinone. Coenzyme Q or ubiquinone receives these electrons from complex 1 and the complex 2, and it acts as a carrier protein or a carrier molecule of this enzyme to complex 3. It donates them to complex 3. Again, we are saying complex 1, when energized, receives the 2 electrons from NADH, gets energized, donates these electrons to coenzyme Q. Then the NADH gets converted to NAD+. In the process, this complex pumps 4 protons from the mitochondrial matrix into the intermembrane space. Then complex 2, also known as the succinate dehydrogenase or succinate dehydrogenase, receives electrons from this compound FADH2, which gets converted to FAD as it donates the 2 electrons to complex 2. So these electrons that are donated to both complex 1 and complex 2 are also donated to ubiquinone or coenzyme Q. This is a carrier protein. So as it receives these electrons, it gets energized and it moves them and transports them to complex 3. So once complex 3 receives these electrons, it gets energized and also pumps protons, 4 protons from the inner mitochondrial matrix into the intermembrane space. So complex 3, once it receives these electrons from coenzyme Q, it pumps 4 protons from the inner mitochondrial matrix into the intermembrane space. So complex 3, once it receives protons from coenzyme Q or ubiquinone, it pumps protons from the inner mitochondrial matrix into the intermembrane space. Then from complex 3, cytochrome C picks up these electrons, gets energized and donates them to complex 4. Cytochrome C picks up these electrons and donates them to complex 4. Once complex 4 receives these electrons, it also gets energized and then it pumps 2 protons into the intermembrane space. So you can see that from these 4 complex proteins that are here, only complex 1, complex 3, and complex 4 are donating protons into the intermembrane space. Complex 1 donates 4. Complex 2 donates 4. Complex 4 donates 2 protons into the intermembrane space. Now remember that these electrons were received by complex 4, and the final electron acceptor now will be what? The oxygen. So this electron acceptor, which is the oxygen molecule, once this oxygen molecule receives these electrons, and in the presence of the protons, the final product is what? Water. 2 molecules of what? Water. That is what happens. Now, remember that in the intermembrane space, there was an aggregation of protons here. You created what is known as the proton motive force, or there's a proton gradient that was created. So this proton gradient that is created is the one now that comes back here to pass through the ATP synthase. So as there is an accumulation here, now you create a proton motive force. So since the gradient has been created, the protons have to come back, and as they pass through this channel here, the as protons are entering back into the mitochondrial space, they are also supplying the energy that is needed to react ADP with the inorganic phosphate to create ATP. So about 3 protons are needed to convert this, and 1 proton is needed to create the energy to drive the protons from from the intermembrane space into the mitochondrial matrix. So 4 protons at a point are required to generate ATP. 4 protons are required. 3 of them are used in the reaction of ADP to with ADP reacting with inorganic phosphate to convert or to form ATP. 3 of them are used, and 1 is used for the movement that is involved here. So that is the electron transport chain. So as the electrons are moving through, ions are pumped into the intermembrane space, creating a proton gradient or a proton motive force. And then protons flow back into the mitochondrial matrix through the ATP synthase complex 5. And this is what drives the phosphorylation of what? ATP to ADP. So remember that ADP was reacting with the inorganic phosphate. Yeah, that is what makes what the process that is known as the phosphorylation. Suppose we are phosphorylating or adding a phosphate group to the inorganic phosphate to ADP. Yeah, this is simply just the same thing there. You can see that you have NADH to complex 1, NAD plus, then you have protons going into the mitochondrial inner mitochondrial membrane space. Then complex 2 there, FADH to FAD, then ubiquinone receives these electrons, transmits them to complex 3. It also pumps electrons. The cytochrome C receives these electrons, pumps them there. Then the final electron acceptor is oxygen, which reacts with the hydrogen to form or give us water molecules. Then because of the proton motive force that is created there, protons begin to fall back into the inner mitochondrial matrix. But as they do that, they also drive the activity of ATP synthase to promote the reaction between ADP and the inorganic phosphate to go forward to create ATP. Yeah, which is simply the same thing here. Yeah, now what is the how much energy or ATP is created? So each NADH that is donated, or each NADH that goes to complex 1, so donates electrons to complex 1, then the electrons move through the chain. And if you remember, NADH, when it enters here, so its electrons start from this point. So it means that by the time these electrons are reaching here, it would have pumped 4 protons at complex 1, 4 protons at complex 3, and 2 protons at complex 4, which means that in total it pumps about 10 protons as it moves from complex 1 to complex 4. FATH, on the other hand, because it bypasses complex 1, meaning the number of protons that are pumped as the result of FADH is what? 6, because it will be 4 that are pumped at complex 3 and the 2 at complex 4. So you have about 6 protons. That is for FADH. So for NADH, we're saying about 10 protons are pumped into the inner intermembrane space. So ATP synthase, that enzyme, requires about 4 protons per ATP. So 3 are used in phosphorylation and 1 is used for transport. So out of this 10, about 2.5 ATPs are produced. Then for FADH2, which donates electrons at complex 2, because it is downstream to complex 1, it means it only pumps about 6 protons. And with 6 protons, it's about 1.5 ATPs per FADH2. So that is the amount of ATP that is produced. So each NADH produces about 2.5 ATP molecules. Then FADH2 produces about 1.5 ATP molecules. So that is one NADH produces 2.5. One FADH2 produces 1.5 ATP molecules. That is the energy generation, or the ATP output. And that marks the end of our lesson. Thank you, guys.